So I brought the teacher from the Student Hub, Akili Joseph. The question that we are doing today is
the May-June 2014 paper, question 2. So let's start. Protein synthesis requires 2 steps, transcription and translation. So table 1 is an incomplete comparison of some features of transcription and translation in eukaryotes. So complete table 1 by writing the correct answers in the relevant spaces in the table. When you have DNA being transcribed into RNA, the RNA then leaves the nucleus and goes into the cytoplasm where the ribosomes are that make the protein. So then where translation would happen where the ribosomes are, which is in the cytoplasm. So here you would put generally in cytoplasm.
okay.
So then the next thing they ask us to compare, the next characteristic or feature they ask us to compare is the precursor molecule, right? So for transcription, remember transcription happens when DNA is transcribed into RNA. So the precursor molecule is the one that's come before, right? Before the arrow, which would be DNA, because DNA is what is transcribed into RNA. So the precursor molecule is going to be DNA. Likewise, they filled in mRNA for us. For translation, it's going from RNA translated into protein. So the precursor molecule, precursor, what comes before, would be RNA or mRNA as they put it, right? Because remember, it's messenger RNA.
Got it.
So that's an easy one. Then it goes enzymes and or factors involved in this. So in transcription, the actual enzyme that does the building of the mRNA is an enzyme known as RNA Polymerase, right? A polymer is just a compound that's made up of many small compounds. So it's going to take the nucleic acids that make DNA and join them together, right? The nucleic acids make DNA and nucleic acids also make RNA. So if there was an enzyme that was making DNA, it would be DNA polymerase. But because this enzyme is making RNA, it will be RNA polymerase.
Nice.
So you can name the enzyme here RNA polymerase. So then they're asking you for the same thing, like what helps the process of translation happen, right? So for translation to happen, there needs to be ribosomes that actually build the amino acids together. So now we're making a different polymer, which is protein, and protein is made up of amino acids joined together. So then what will be joining the amino acids together would be the ribosome that's actually made up a different type of RNA known as rRNA or ribosomal RNA. So we can put that here, that rRNA is involved in joining together the amino acids. Another one we can talk about here would be for the ribosomes to be able to join the amino acids together, something has to bring the amino acids to where the ribosomes are. And what brings those amino acids would be a different type of RNA, again known as tRNA, and that is transport RNA, right? Or transfer RNA, right? So they are just like, your little errand boys that will fetch the right amino acids and bring it for the ribosomes to be able to incorporate the correct one, right? So you can then talk about, you can have tRNA involved there, and then like how tRNA works and how we're going to talk about it in more detail just now is that there are enzymes that actually load the amino acid onto the tRNA. So then that would be tRNA transferase. So what I'm showing you here is that any one of these answers would have given you your mark here, but there are multiple correct answers that can go into this box because they're asking you for any enzyme and or factors that would be involved in the process, right? So there are multiple, once you understand how the process works, any of those aspects or factors that you list here, you will get your 1 mark, right?
Got it.
Cool. And then lastly, they ask us about the function, right? So let's go with the one they gave us. They gave us the function of translation, and they say translation produces the peptide sequence, which is complementary to the mRNA, right? So the peptide sequence, that's the sequence of amino acids. So that's what peptide sequence is, the sequence of amino acids, which is complementary to the mRNA. So remember, the mRNA leaves the nucleus and comes into the cytoplasm where the ribosomes are. So it's producing the peptide sequence, which is complementary to the mRNA molecule that would have left the nucleus.
Right.
But before we can get that mRNA molecule, that's where transcription has to come in, right? Because DNA has the instructions about how to make the protein. A copy of those instructions is converted into the RNA molecule, the messenger RNA molecule, and then that messenger RNA molecule leaves the nucleus, goes to the ribosomes and tell the ribosomes what to do, and the ribosomes read the mRNA and make the appropriate peptide sequence that will become your protein, right? So that's the overall process that's happening. So what would be the importance of transcription? It is that you want to make mRNA that is complementary to DNA. So you have DNA, you make a molecule mRNA that's complementary to the DNA because you want to get that message out. The mRNA is able to leave the nucleus, go to the ribosomes, and then you want your ribosomes to make a protein or a peptide sequence that is complementary to the mRNA.
Got it.
And that's how we get the protein that we need, right? So just lastly, to complete the table, we would say that the function of transcription is to produce mRNA, which is complementary to our precursor molecule there, which would be DNA.
All right, so the protein is a peptide sequence.
Yes.
And the mRNA is what?
It's a type of nucleic acid. So where amino acids are joined together to form proteins, you have nucleic acids being joined together to form RNA.
Got it.
This part of the question, 2a part 1, was with 4 marks. So we get our 1, 2, 3, 4. We Boston and CXC. We're moving on to the next part of the question now, right?
All right.